Hello, I'm Professor Sims, and in this video I will discuss the lymphatic and immune systems. This is the fifth in a series of 10 lessons held as part of my Anatomy and Physiology 2 online laboratory course. If you're a student currently enrolled in this course, please consult the syllabus and course Moodle site for assignments, quizzes, due dates, and other course information. By the end of this lesson, you will be able to locate and identify major lymph vessels and organs, describe the structure and function of primary and secondary lymphatic organs, discuss the cells of the immune system, how they function, and their relationship with lymphatic systems, identify the specific cells of the immune system, differentiate between innate and adaptive immunity, and demonstrate how an ELISA test detects disease-specific antibodies. The lymphatic system is a very important component of both fluid regulation and immune defense. One of its key functions is to return excess interstitial fluid, about 3 liters a day, back into the bloodstream. And without this function, fluid would accumulate in tissues and cause swelling or edema. This system also maintains fluid balance, helps transport dietary lipids, and serves as a transport network for immune cells, especially the lymphocytes. It's responsible for filtering pathogens and circulating immune components throughout the body. You can see in this diagram here that the lymph originates in the lymphatic capillaries, which collect fluid from tissues. And then that fluid, now called lymph, moves into collecting lymphatic vessels, which are equipped with valves to ensure one-way flow, similar to what we saw in the veins. Then from there, the lymph moves through the lymph nodes, where immune surveillance occurs. Then it travels through larger lymph trunks and into the right lymphatic duct, which is also called the thoracic duct. The right lymphatic duct collects lymph from the right upper limb, right side of the head, and right thorax, and drains it into the right subclavian vein. And then this same duct, the thoracic duct, handles everything else, like draining the lymph from the left side of the body and lower limbs into the left subclavian vein. Note how the lymphatic and circulatory systems are closely integrated, the lymphatic system acting like a bridge, returning fluid to the bloodstream while also supporting immune function throughout the body. Take a moment to trace the structures in the diagram, and you'll be able to see the close relationship between the arterial system, venous system, and lymphatic vessels, as well as the directional flow through nodes, trunks, and ducts. See how it is. It all starts with blood plasma, which is the fluid portion of blood, and it filters out the capillaries into the surrounding areas, or the surrounding tissues. Once outside the vessels, this fluid is called interstitial fluid, and it surrounds the cells, delivering nutrients and removing waste. But not all of this fluid is reabsorbed directly into the blood. A portion of it, which is about 3 liters a day, enters lymphatic capillaries, and at this point the fluid, again, is named lymph. Now, lymph flows through a series of lymphatic vessels, which are structured similarly to veins and also include the one-way valves to maintain forward flow. And then along the path, lymph is directed through lymph nodes. Inside each lymph node, lymphocytes, which we know are a type of white blood cell, monitor the fluid for pathogens, debris, and abnormal cells. And then if anything harmful is detected, an immune response is initiated. This is an important part of how the body prevents infections from spreading. After filtration, the cleaned lymph travels through larger lymphatic vessels, essentially collecting into the right lymphatic duct or thoracic duct, depending on which region of the body it came from. Finally, lymph is returned to the bloodstream by draining into the right or left subclavial veins, rejoining circulation and completing the cycle. This continuous process helps maintain fluid balance, supports immune defense, and keeps tissue environment stable, which makes it, again, a really important part of your body's homeostasis and protection. So let's take a closer look at the key structures of the lymphatic system and how they function. So we start at the lymphatic capillaries. Those are tiny, thin-walled vessels located throughout the body's tissue, and their main job is to absorb excess interstitial fluid. Remember, the fluid that surrounds around cells and then bring it into the lymphatic system where it becomes lymph. And then from there, the lymph flows into larger lymphatic vessels, and these vessels are equipped with one-way valves to keep the lymph from moving backward. It keeps them moving forward even against gravity. And then as lymph travels, it's directed through the lymph nodes, which kind of act like security checkpoints. Lymph nodes are small, bean-shaped structures filled with lymphocytes and macrophages, and they need those to filter the lymph, detect and respond to pathogens like bacteria and viruses, and of course remove damaged or abnormal cells. Clusters of lymph nodes are found in areas like the neck, the armpits, and the groin, places where the body is most vulnerable to infection. Now let's talk a little bit about bone marrow as well. Shown in the diagram near the pelvis and long bones, this is where the lymphocytes are produced, and it's also the site of B cell maturation. B cells play an important role in adaptive immunity by producing antibodies. And then also visible in the diagram are like the tonsils and adenoids in the head and neck that guard against inhaled or ingested pathogens. The thymus, located in the chest, is where the T cells mature, and the spleen filters the blood, removing old red blood cells and detecting blood-borne pathogens. The lymphatic ducts return filtered lymph back into the venous system through the subclavian veins. Let's zoom in on some specialized lymphatic organs and tissues that provide targeted immune protection in specific regions of the body. We start with the tonsils, located in the throat. These are some of the first lymphatic tissues to encounter pathogens that are breathed in or swallowed. Positioned around the entrance to the respiratory and digestive tracts, tonsils act like early warning detectors, capturing and responding to microbes before they can spread. Next is the thymus, located just behind the sternum. While we already mentioned its role in T cell maturation, it's also important to remember that the thymus is most active during childhood. It gradually shrinks with age, but it plays a key role in shaping the immune system early in life. Then you have the spleen, found in the upper right part of the abdomen. The spleen performs a different function than the lymph nodes. It filters the blood directly, not the lymph, and it removes the red blood cells, stores white blood cells, and again, it helps respond to blood-borne infection. And then next we have the Peyer's patches, which are clusters of lymphoid tissue embedded into the wall of the small intestine. These monitor the gut for pathogens and help regulate immune response to what we eat and the microbes that we naturally carry in our digestive tract. Then finally have the appendix, located just off the large intestine, and the appendix contains lymphoid follicles similar to those in the Peyer's patches. While it was once thought to be vestigial, we now have come to understand that the appendix plays a role in gut immunity, especially during early development. So all of these structures extend immune surveillance beyond the bloodstream and into the respiratory, digestive, and circulatory systems, helping the lymphatic system protect the entire body. So let's break that down into three main lines of defense. The first line of defense is physical and chemical barriers. This kind of protection is primarily the skin and mucous membranes that act as a wall to keep pathogens out. But then beyond that, we also have specialized immune tissues embedded within these barriers. For example, MALT, or mucosa-associated lymphoid tissue, is found in mucosal linings of the gut, lungs, urinary, and reproductive tracts. It provides localized immune surveillance where pathogens are most likely to enter. And then a subtype of MALT is called BOLT, or bronchus-associated lymphoid tissue, located in the lungs, which of course is an important checkpoint for inhaled pathogens. If a pathogen gets past these barrier defenses, the second line of defense kicks in, the innate immune system response. This response is rapid but non-specific, meaning it doesn't target a particular microbe. Instead, it reacts to general signs of infection or damage. Key players here include phagocytes like macrophages and neutrophils that engulf invaders, natural killer cells cells that destroy abnormal cells, and inflammatory cells that trigger swelling, redness, and heat to limit spread and attract more immune cells. And then if the innate response isn't enough, the body activates the third line of defense called the adaptive immune response. And this is going to be slower to initiate, but highly specific. B cells produce antibodies that target and neutralize specific pathogens, while T cells help coordinate the immune response and directly kill infected cells. The lymphatic system ultimately returns fluid to venous circulation, and this is done through two main ducts, the right lymphatic duct and the much larger thoracic duct. On the right side of the body, the right lymphatic duct drains lymph from the upper right quadrant of the body, including the right side of the head and neck, right arm, and right side of the thorax. It empties into the right subclavian vein, where it joins the right internal jugular vein. The rest of the body, which is most of it, is drained by the thoracic duct. This duct begins in the cisterna chyli, which is a sac-like structure located in the abdomen. From there, the thoracic duct ascends through the thoracic cavity and eventually drains into the left subclavian vein, where it meets the left internal jugular vein. So to summarize, right lymphatic duct equals upper right body drains into right subclavian vein. Thoracic duct equals everything else, and it drains into the left subclavian vein. These ducts are very important for maintaining fluid balance and ensuring that the lymph is filtered and returned to the bloodstream for circulation. This figure here illustrates the fluid balance, that is how fluid moves in and out of capillaries, and how the lymphatic system helps recover any excess fluid. On the left side, near the arterial end of the capillary bed, we see a process called filtration. The net filtration pressure here is plus 10 milligrams per mercury, meaning that the pressure inside the capillary, called the capillary hydrostatic pressure, is 35 milligrams per mercury, and that's greater than the blood colloidal osmotic pressure, which is only 25 mmHg. This pressure difference pushes fluid out of the capillaries and into the surrounding tissue. Now as we move to the middle, the capillary pressures equalize. The hydrostatic pressure that has dropped to 25 mmHg, matching the osmotic pressure. And this results in no net movement of fluid. It's a balance point. Then at the venous end, we reach the process of reabsorption. Here the capillary hydrostatic pressure has dropped even further to 18 milligrams of mercury, while the blood colloidal osmotic pressure remains at 25 mmHg, and this creates a net filtration pressure of negative 7 mmHg, drawing the fluid back into the capillary. Let's take a closer look at how interstitial fluid becomes lymph and enters the lymphatic capillaries. So in this diagram, we can see an arteriole delivering blood into a capillary bed where fluid filters out into the surrounding tissue. This fluid, now called interstitial fluid, bathes the tissue cells, delivering oxygen and nutrients. And not all of the fluid is reabsorbed by the venules, and the remaining fluid still surrounding the tissue is taken up by the lymph capillaries shown here. Notice the structure of these capillaries. They're made of overlapping endothelial cells that form flap-like valves. When interstitial pressure increases, these flaps will open inward, allowing fluid to enter, and then once inside, the fluid is now called lymph, and the endothelial flaps act like one-way valves. They close if the pressure tries to force fluid back out, preventing backflow. This is aided by anchoring collagen fibers, which hold the lymphatic vessels in place and help maintain the opening of the capillary. Also shown are larger lymphatic vessels that transport lymph away from the tissue and eventually return it to the bloodstream. Lymph nodes are small bean-shaped organs, typically less than an inch in size. Each node is responsible for draining lymph from a specific region of the body. On the outside, each node is encased in a fibrous capsule that protects the internal tissue and helps define the node's shape. Inside, the node is organized into two main regions, the cortex and the medulla. The cortex, located near the outer edge, contains clusters of lymphoid follicles, which house B cells and T cells, the lymphocytes that drive the adaptive immune response. When an infection is detected, these follicles can become enlarged and activated, forming what we call a reactive lymph node. At the center of the node is the medulla, which contains medullary cords and macrophages. These macrophages play a key role in engulfing and digesting pathogens, then presenting fragments to lymphocytes to initiate a more targeted immune response. Lymph enters the node through the afferent vessels, then flows through a series of internal sinuses, small channels that act like filters, before exiting via efferent vessels. This slow, guided flow allows immune cells time to detect and respond to any invaders. This figure shows the locations of key lymph nodes and lymphatic structures throughout the body. If you start at the top in the neck, you find the cervical lymph nodes, which help to monitor lymph coming from the head and neck. These are often the ones you feel swell when you're fighting a cold or a sore throat. Moving downward, you have the axillary lymph nodes. These are located in the armpits. They drain lymph from the upper limbs, chest wall, and breast, including the lymphatics of the mammary glands, and they're especially important in breast health and cancer screenings. In the abdominal region, we see the cisterna chyli, which is here. It is a sac-like structure at the base of the thoracic duct that collects lymph from the lumbar and intestinal trunks and drains organs in the lower abdomen and pelvis. Next, the lumbar lymph nodes line the lower back and help filter lymph from the kidney, adrenal glands, and other retroperitoneal structure. Pelvic lymph nodes lie deeper in the pelvis and receive lymph from reproductive and urinary organs. And then the inguinal lymph nodes are located in the groin, and they filter lymph from the lower limbs, external genitalia, and lower abdomen wall. You might notice these swell if there's an infection in the legs or the feet. Also shown are the lymphatics of the upper and lower limbs, which return fluid toward the thoracic duct or right lymphatic duct, depending on the side of the body. You can also see the thymus and spleen in this figure, which are, of course, essential lymphatic organs. The thymus, located above the heart, that's where the T cells mature. And the spleen in the upper left abdomen is where the blood is filtered. In the roof of the nasopharynx, you can find the pharyngeal tonsil. This is sometimes referred to as the adenoid, especially when swollen in children, and it plays a defensive role by monitoring inhaled pathogens. The palatine tonsil, those are the tonsils that are located on either side of the pharynx between the palatoglossal and palatopharyngeal arches, and these are the ones most commonly removed in a tonsillectomy. In this diagram, these tonsils appear very swollen and inflamed, and this is usually due to infection. Then at the base of the tongue is the lingual tonsil. Although these are less prominent, they also contribute to immune defense, particularly against ingested pathogens. And now on the right side over here, you have a histological micrograph of the palatine tonsil. Notice the stratified squamous epithelium, which protects the surface from friction and potential pathogens. And then beneath the surface, the tonsil contains multiple crypts, which are invaginations that trap and expose pathogens to immune cells. Within the tissue, the germinal centers can be seen. These are regions rich in B lymphocytes actively producing antibodies. And together, the tonsils form the first line of immune surveillance in the throat, responding rapidly to pathogens that enter through the mouth or nose. The thymus here, just above the heart, is nestled in the upper thoracic cavity behind the sternum. It lies near other lymphatic structures, including the adenoids, tonsils, spleen, and lymph nodes, all visible on the diagram for context. The right lymphatic duct entering the venous circulation is also shown, reminding us of the close relationship between the lymphatic and circulatory systems. Now, if you look at the histological section of the thymus, you can clearly see lobular organization separated by trabeculae. Trabeculae are bands of connective tissue extending from the fibrous capsule, and each lobule contains two distinctive regions. Cortex appears darker due to the high density of developing thymus thymocytes, which are immature T cells. The medulla is lighter in color. I hope you can see that here. It's lighter. The medulla contains fewer thymocytes as well as medullary epithelial cells, macrophages, and dendritic cells, all of which contribute to the process of T cell maturation and what is known as self-tolerance testing. The cortical epithelial cells support and guide the development of thymocytes, while macrophages help remove those that fail to mature properly. Dendritic cells are involved in antigen presentation, which is an important step in immune response. Blood vessels running through the thymus are also shown, emphasizing how immune cells and signals are transported into and out of the organ. And then, as the primary site for T cell maturation, the thymus is most active during childhood, and it begins to shrink or involute with age. This structure-function relationship is critical to understanding how the adaptive immune system is built early in life. The spleen is located in the upper left quadrant of the abdomen, just beneath the diaphragm, and it is attached to the stomach. It connects to the circulatory system via the splenic artery and splenic vein, both of which enter and exit at the ileum, which is the central indentation on the spleen's surface. Here's the ileum here, the splenic artery here, and the splenic vein. It's very small, but you can see the little blue spot right there. Histologically, the spleen contains two major types of tissue, the white pulp and the red pulp. The white pulp shown here and the red pulp out here. The white pulp surrounds a central artery or arteriole and contains germinal centers, which are rich in lymphocytes. This region functions like a lymph node, filtering blood for pathogens and initiating immune responses. The marginal zone, which encircles the white lymph, acts as a transition area. It sequesters particulate antigens from the bloodstream and presents them to immune cells in the white pulp, kickstarting an immune response. Now, red pulp is the area responsible for filtering and recycling red blood cells, and it consists of a network of venous sinuses and splenic cords, or trabeculae, where old or damaged red blood cells are removed from circulation. Here you can also see the arterial capillaries, arterioles, and venules. There's a venous sinus here, venule here. These all help to distribute blood through both white and red pulp for efficient immune surveillance and filtration. Peyer's patches are clusters of lymphatic nodules embedded in the submucosa of the ileum, which is the final portion of the small intestine. Their primary role is to capture and destroy bacteria, helping to prevent foreign pathogens from crossing the intestinal wall and entering the bloodstream. So let's walk through the structures labeled in the diagram to understand how these nodules are situated within the intestinal wall. At the innermost surface, we have villi. Villi are finger-like projections that increase the surface area for nutrient absorption. And then within each villus is a lacteal, which is a small lymphatic vessel that absorbs dietary fats. Beneath the epithelium lies the lamina propria. This is a connective tissue layer containing intestinal glands and immune cells. Then below that, you can see the muscularis mucosa, this layer here. This is partially disrupted where the Peyer's patches expand into the mucosa. The lymphatic nodules of Peyer's patches are clearly visible in the submucosa. These nodules contain densely packed immune cells, including B and T lymphocytes, which monitor the contents of the intestinal lumen and launch immune responses as needed. And then surrounding these are the layers of the intestinal wall. The muscularis externa is composed of an inner circular layer and an outer longitudinal area of smooth muscle, which generate peristaltic movement. And then the outermost layer is the serosa, or visceral peritoneum, which helps anchor and support the intestine. All blood cells originate from a common source, the multipotent hematopoietic stem cell, also known as the hemocytoblast. From this single stem cell, two major progenitor lineages emerge. The myeloid lineage, which gives rise to erythrocytes, the red blood cells, responsible for oxygen transport, basophils, which play a role in allergic reactions and inflammation, neutrophils, the most abundant white blood cells and the first key responders to infection, eosinophils, which combat parasites and participate in allergic response, monocytes, which circulate in the blood and differentiate into macrophages and dendritic cells. Then the lymphoid lineage produces B lymphocytes, which mature in the bone marrow and produce antibodies, T lymphocytes, which mature in the thymus and coordinate immune responses or kill infected cells directly, natural killer cells, which are part of the innate immune system and destroy virus-infected or tumor cells without prior sensitization. And this branching tree structure from a single pluripotent stem cell to a variety of specialized immune and blood cells is fundamental to immunity and homeostasis. The bone marrow is constantly adjusting the production of these cells based on the body's needs, whether it's responding to infection, replacing lost cells, or maintaining daily physiological balance. Now here we are zooming in specifically on the lymphoid branch. In this first column here, you have the type of lymphocyte, B, T, plasma, or NK. The second column indicates that all are classified as white blood cells. And then the third column outlines their primary functions. We just went over these. The B cells mature in the bone marrow. They make antibodies. They recognize antigens and initiate humoral immune response. T cells mature in the thymus, and they're involved in cell-mediated immune response. Subtypes include the helper T cells, which coordinate immune system, cytotoxic T cells, which kill infected or abnormal cells. Then you have natural killer cells. Again, they're a part of the innate immune system. They recognize and destroy cells that are infected or cancerous without the need for prior antigen exposure. You can trace all of these back to the stem cells in the bone marrow, which first divide into myeloid and lymphoid progenitor cells. Now, although platelets are not lymphocytes, the diagram includes them just to emphasize that all blood and all immune cells ultimately come from the same origin. Now let's discuss the 5 major classes of antibodies, also known as immunoglobulins, that each play a distinct role in immune response. So let's begin with IgM. IgM is the first antibody produced when the body encounters a new antigen. It's especially good at forming antigen-antibody complexes, which helps initiate immune defense quickly, and its large size makes it effective at neutralizing threats early in immune response. Next is IgG. This is the most abundant antibody in the bloodstream. It provides long-term immunity and is the only antibody that can cross the placenta, which gives passive immunity to the fetus. If you've ever had a vaccine or recovered from an infection, it's IgG that helps you to stay protected in the long run. IgA is found primarily in mucosal secretions like saliva, tears, and breast milk. It helps to protect mucosal surfaces such as the gastrointestinal, respiratory, and urogenital tracts where pathogens are very likely to enter the body. Then we have IgE. IgE is central to allergic reactions and defense against parasitic infections. It's normally present in very small amounts, but it can trigger strong immune responses, sometimes overly strong as seen in some of the allergic symptoms. Then IgD, it's the least understood antibody. It's primarily found on the surface of immature B cells, where it acts as a receptor for antigens. While its exact function in immune activation is still not quite understood, it appears to be involved in initiating B cell activation. Let's talk now about one of the most important tools in clinical and research into immunology. So this is ELISA. ELISA stands for Enzyme Linked Immunosorbent Assay. ELISA is a gold standard plate-based assay that allows us to detect and quantify a wide range of biological molecules, most commonly antibodies, but also antigens, proteins, and hormones. It's widely used due to its high sensitivity and specificity, making it extremely reliable for diagnosing conditions such as HIV, hepatitis, and even pregnancy. And it plays a really important role in detecting tumor markers like PSA and CEA. So how does it work? ELISA relies on antigen-antibody interactions, a very specific binding process. And then once binding occurs, an enzyme-linked antibody is introduced, and it triggers a color change in the presence of a substrate. The intensity of that color directly corresponds to the amount of the target molecule present. Then we can measure that color using a spectrophotometer or a plate reader to determine concentration. There are several types of ELISA. You have direct ELISA, where the primary antibody is directly linked to the enzyme. Indirect ELISA, which uses a secondary antibody for signal application. Sandwich ELISA, where the target antigen is sandwiched between two antibodies. This is excellent for large proteins or really complex samples. And competitive ELISA, where the sample competes with a labeled antigen. This is useful for small molecules and when high sensitivity is required. So in summary, ELISA is a powerful diagnostic and research tool, and understanding the principles of ELISA is going to help you grasp how laboratory tests work to detect immune responses. So a positive ELISA result is indicated by wells that show a strong color change. Usually, it starts clear and then it usually goes to a deep yellow or orange color. This means that the target antibody or antigen is present in the sample at a detectable level. In contrast, a negative result is shown when the wells remain clear, or like here where they only have a very light color. This suggests that the target molecule is absent or below the detection limit, at least. ELISA can be used in two ways, qualitatively or quantitatively. In a qualitative test, the presence or absence of color simply tells us whether the target is detected or not. In a quantitative test, the intensity of the color is measured and compared against a standard curve of known concentrations using a spectrophotometer, and this allows us to determine the exact amount of the target molecule is in each of these wells. This here is a typical 96 well plate after an ELISA test was run. Each well corresponds to a patient sample or a standard. So these B, C, D are all patient samples, and A is the negative control, the standard. And you can see how wells with the darker yellow or orange hues are observed, and those indicate higher concentrations of the antibody that we're looking for. For your pre-lab activities, please read the Lymphatic and Immune System chapter in your textbook. That's chapter 21 in OpenStax. Then review the introduction and background sections in chapter 5 of the lab manual. Of course, this outlines the objectives and procedures, and then complete the pre-lab exercises 5.1 through 5.4 and use the answers from those exercises to complete the lab 5 quiz on Moodle. For lab exercise 5.1, you're going to work with lymphatic system models, or for your online students, you can use figures 5.4 through 5.6 in the lab manual. The task is to identify and label key structures, including the cervical lymph nodes, axillary lymph nodes, right jugular trunk, lymphatic vessels, right lymphatic duct, thymus, heart, and bone marrow. Take your time so you know where these structures are positioned and how they connect within the rest of the lymphatic system. So here you have an anatomical model that has the lymph nodes and spleen, cisterna, chyle, thoracic duct, the cervical lymph nodes, auxiliary lymph nodes. These are all labeled here. Then you have the afferent lymphatic, the capsule, the cortex, the efferent lymphatic, the lymphatic valve. You can see here, there's another one here, but it's not labeled. Medullary sinus, these capillaries here, medullary cord, and then the germinal center. This is a model here that is showing the 3 tonsils that we discussed here. And then another model here is showing the thoracic duct. Then for lab exercise 5.2, this is the histological examination. So we will be using slides in lab, but online we're using figures 5.7 and 5.8. You want to identify medulla, cortex, diffuse lymphoid tissue, follicle, capsule, septum, lymph node, cortical sinus, and subcapsular sinus. So the lymph nodes are encapsulated and they show clear structural organization. The cortex contains lymphoid follicles, predominantly B cell areas, often with prominent germinal centers where activated B cells proliferate. Surrounding the follicles is the paracortex. here, rich in T cells. The medulla, you can see that here, it's lighter in color. It contains medullary cords filled with plasma cells and B cells, as well as medullary sinuses, right? The spaces where the lymph flows and is filtered. Then under the scope, you will also notice a fine reticular framework supporting the cells. Reticular fibers made of type 3 collagen create a scaffold, best visualized with silver stains. This here is just a close-up showing the difference between the cortex area and the medulla area, and then the follicles within the cortex. When viewing the thymus, look for its distinctive lobulated structure surrounded by connective tissue capsule. Each lobule has a dark staining cortex densely packed with immature T lymphocytes, again, thymocytes. These cells give the cortex a solid basophilic appearance, this deep purplish blue appearance. Scattered epithelial reticular cells and macrophages are also present here. And then the medulla, again, you can see that as a lighter staining and less dense structure containing fewer lymphocytes than the cortex. Key identifying features of the medulla are the Hassel's corpuscles, which are concentric eosinophilic epithelial aggregates that are unique to the thymus. So the little pink tint that you see in here, those are epithelial aggregates. Now remember, the thymus lacks lymphoid nodules, which helps to distinguish it from other lymphoid tissues. And then the spleen is even more different from lymph nodes because it lacks a defined cortex and medulla. Instead, it has white pulp and red pulp. The white pulp appears as circular or oval lymphoid areas surrounding central arteries. Within the white pulp, you can identify the periarteriolar lymphoid sheath, which is rich in T cells. This one's not quite zoomed in enough for you to see that. And then adjacent follicles rich in B cells. Again, we're not zoomed in enough to see that here, but just remember the white pulp is going to have a lymphoid sheath rich in T cells and adjacent follicles rich in B cells. And then the red pulp surrounds, so all of this is red pulp, surrounds the white pulp and consists of splenic sinuses and cords. This is the area that filters the blood, right? Look for the trabecule connective tissue, and this here is the trabecular artery here. Now, we talked about Peyer's patches. This is how it looks under the microscope here. They look like, well, they are lymphoid nodules beneath the intestinal epithelium, and the overlying epithelium often shows specialized M cells involved in antigen sampling. The key layers here are going to be the surface epithelium, lamina proxima, and disrupted muscularis mucosa near the patches. So these are the patches here, and this is the villi. We looked over all of these structures in the diagram earlier in the presentation. This is a cross-section here of the patches showing the mucosa layer here, Peyer's patches here. Now, finally, for lab exercise 5.3, the ELISA testing interactive web activity. So first you want to navigate to this site here. Then the virtual lab is divided into 4 main stages that you will complete step by step. Make sure you go through the introduction where you will get an overview of ELISA testing and how it's used to detect antibodies. Then you will do a blood sample analysis where you're going to practice preparing and analyzing blood samples for testing. You're going to do the placing and preparing of slides. You're going to pipette samples into wells. You're going to follow incubation steps. These are images showing examples of what it will look like. You'll have to click on the pipetter, pull from the serum, pipette different buffers. You will go through all of the steps just as if you were in the laboratory. You will have to interpret your test results to make clinical decisions. This activity, the point is to reinforce your understanding of the immunological testing, the ELISA testing, and prepare you for practical applications in clinical and research settings that you will have later in your academic career. And again, we'll back up to the 4 steps. While you're doing this activity, you're working through these stages, and you will have to follow the prompts step by step and observe how these samples react. And if you're ever lost, you just want to click through these tabs up here. It even has a glossary. If you're not sure how to do what you're supposed to be doing, it even has a help section that can show you how to do things. The notebook part is keeping track of all of your observations and everything that you did to get there. Okay, so thank you guys for watching, and don't forget to do the reading. Check the description for more videos related to these topics, and leave your questions for me in the comments below.